Thank you for that introduction. I'll be talking today about best practices related to determination of stability of drugs, both from a development and a regulatory perspective. When I use the word drugs, I'm intending to mean both drug substance and drug product. You may be looking at the title slide, which says stability. Why do we care? Justifying your product. And saying to yourself, well, that's an easy question to answer. We want to know how long is the product good for? And that's a good short answer. However, there is a lot of planning that goes into determining that answer. So far as justifying your product, the presentation is intended to provide the available tools and ideas for use in providing that justification. Here's an outline of the presentation. There will be an introduction, which is just a brief background. Expectations, which are about the program itself. Considerations, the considerations are for developing the program. Guidance, the guidance that is typically used in an assessment for stability. The guidance content, which is just touching on the major topics in the various guidances. And then some final thoughts about the presentation and going forward. Well, why do we care? We care because we have unknowns in our system. At product release, we know the status of the product. The product is acceptable or it would not be released. However, once it's released, there are several unknowns. One of those unknowns is how long it is before the patient takes the particular drug. That's related to the distribution scheme, the time in the market, the time in storage, ownership by the patient. Another unknown is what is the environment that the drug experiences. It's in the distribution and it's in sometimes uncontrolled environmental situations. So when the patient actually uses the drug, we're not entirely sure whether or not the drug is still acceptable for the patient. A stability program is a rationally designed data collection program. The data that's collected is describing product characteristics over time. These are intended to help us remove the uncertainties from the previous slide. The result of this data collection is a stability profile. And since this profile incorporates product characteristics, it can be considered to be a product quality measure. There are several expectations regarding stability programs. And one of these expectations is that there's a focus about what a program is designed to show. This is both a regulatory and a development expectation. it should be a protocol with a rational design that will allow you to evaluate the data that comes out of the end of the program. It also should address what changes occur or can be expected to occur, rather than just do testing and seeing what happens. And it also needs to address what is important to the product. For our purposes, what's important to products are the efficacy of the product, the safety of the product, the quality of the product, which incorporates sometimes various performance elements. So what is important to efficacy, safety, and performance? Well, efficacy is in part determined by drug substance content. So, because that addresses whether or not the product might actually help the patient who's taking it. Safety is important because of degradation products and in the product, because they may reach some toxic level or have some deleterious health effects. Performance, or sometimes you might call it quality, might be drug substance availability, because if the drug substance isn't available to the patient's metabolism, then it's not going to be as effective as it's expected to be. And also there are physicochemical properties that sometimes will be useful as an important factor. What is important may be product specific. An example would be some physicochemical properties for different kinds of products. Tablet hardness in itself is not usually considered a requirement for a stability program. However, a chewable tablet hardness is critical to its usability for the patient. So for a chewable tablet, tablet hardness might be expected to be part of the stability program. For an unbuffered solution, pH would be important because of potential pH changes in the unbuffered solutions that might be deleterious to the drug substance content. For products like ophthalmic solutions, which in part depend on contact and residence time on the surface of the eye, of course viscosity would be an important parameter, whereas for some other liquid products, viscosity might not be important to the performance of the product. Among other expectations that one would find from a regulatory standpoint is it would be expected that the ICH Q1 guidance recommendations will be followed. I've said Q1 and not Q1A because there are several Q1 documents regarding stability. Other ICH guidance may be applicable, and all that means is that there may be guidance that affects what goes into the program, what goes into the stability profile. USP/NF, Pharmacopoeia National Formulary requirements, are applicable, because the USP is a U.S. official standard, and it contains monographs and chapters and specifications for drug substance and drug product, and the chapters also address various requirements for treatment of drugs and applicable specifications. What kinds of things might affect these important product properties that we've talked about just generally? Several come to mind. The major ones would be formulation or component interactions, the effect of light, the effect of temperature, because most things would degrade at elevated temperatures, or the product may not maintain its integrity under freezing conditions. Another effect might be chemical degradation, and this would be hydrolysis, reaction with acid and base for, again, chemical degradation. Analytical capability is important because we need to be able to see what changes occur and the extent of the changes. A discussion of what the change is that is expected is also important, because if you know what the change is, the expected change is, then you should be able to figure out what kind of test to do and what kind of material or analyte that you are testing for. Stress studies, at least for drug substance, are very useful for this because they provide stressed or extreme conditions to help generate degraded drug substance. That provides more material to be tested and provides it in a shorter time frame. So this is a discussion also on the sample generation of what you're going to test for. And again, in the case of drug substance, a stressed sample of drug substance would give you a better idea of what likely changes there may be. You also need to consider appropriate test methods and valid test methods. This is not the same as, do you have validated methods, even though people are expected to have validated methods to justify the validity of their analytical results. But what we're talking about here is the test that you're running valid for what you're testing for? Would you expect to see a certain kind of change using a certain kind of test? An example might be, if you're using HPLC, would you expect the changes to have a lot of very fast eluting materials, which you might not see at all in the front of the chromatogram? This is just a list of guidance that relate directly or indirectly to stability issues. I'm not going to go into any detail because we would need the rest of the conference to cover them simply because of the wide scope and the granularity of the information in these guidances. However, I will be highlighting the major topics later in the presentation. First, we have the ICH document Q1A-R2, which is stability testing of new drug substances and products. We have ICH Q1B, which is stability testing, photo stability testing of new drug substances and products. We have Q1C, which is stability testing for new dosage forms. And we have Q1D, which is bracketing and matrixing designs of new drug substances and products. Q1E is a document describing evaluation of stability data. We have guidance for industry, for ANDAs, which is stability testing of drug substances and drug products, question and answers. And we have the United States Pharmacopoeia National Formulary, which is a wealth of information. We have various scale-up and post-approval changes documents, such as SUPAC-IR, which is immediate release solid oral dosage form scale-up and post-approval changes, chemistry, manufacturing and controls, in vitro dissolution testing, and in vivo bioequivalence documentation. There's a guidance SUPAC-MR, which is a modified release solid oral dosage form scale-up and post-approval changes with also chemistry, manufacturing controls, etc. Then there is a SUPAC-SS, which is a non-sterile semi-solid dosage form scale-up and post-approval changes, again with that litany of chemistry, manufacturing controls, in vitro dissolution, and in vivo bioequivalence documentation. There are guidance that are not directly stability guidances, and these actually address product criteria that would affect the protocols and stability program. ICH Q3A through Q3E address impurities, and this is important to stability because those guidances describe controls and descriptions of limitations in the criteria. Basically, they talk about how you set a limit or criteria for various kinds of impurities. ICH Q6A and Q6B are documents describing specifications. They talk about dosage form tests and general versus specific test requirements for different kinds of dosage forms. You may have noticed that there aren't very many FDA-specific guidances on drug stability. This is because FDA has adopted the harmonized ICH guidance for use in application assessment. Here is a series of guidance highlights, which I mentioned a few minutes ago, which again, there's not a lot of detail on these guidances because the guidances are lengthy, but just the major topics that are addressed within these various guidances. Q1A is what we would like to talk about as the basic stability guidance. It contains batch recommendations such as the use of multiple batches. It describes permissible or expected batch scale values. It provides storage conditions for different types of products, freezer, refrigerator, room temperature, to provide standard conditions across the regions during the testing so that products can be compared and match to some extent the local environments of the various regions. It also describes a study commitment, which is a commitment to continue certain kinds of stability testing in the program. And this is to relate market product to application data. In other words, continuation of the study for different size batches or perhaps different other differences to allow an evaluation of whether that continued data collection matches what was used to approve an application. It also includes test frequency, which is a discussion of time intervals between testing and also extent of testing, meaning the duration of the program itself. ICHQ1B photostability testing is, as it says, testing for sensitivity to light. And it's a stress test, and it provides standard conditions for testing of drug products and drug substances. One of the primary uses for this information is as a guide in developing packaging to protect product from the effect of light. ICHQ1C, new dosage forms, is not strictly speaking about new dosage forms. It's about new dosage forms by an owner of an existing application. So this guidance is primarily useful for someone who has an existing application and wants to change some parameter of that existing product to produce a new product, a new dosage form. The guidance says to follow the principles of Q1A, which is again back to the parent or base document. And there is a potential in Q1C for a reduced database on submission of the new dosage form application. Basically what this means is that you may be permitted to submit a reduced data package based on the previously submitted data for the other product. The key thing, I think, for Q1C is to remember that it doesn't apply to anybody who wants a new product. it applies to a change in a product for someone who already has that product. Q1D is a bracketing and matrixing document, and it's concerned with describing and discussing reduced designs for stability protocols. It is concerned with a reduction in the extent of testing. Bracketing and matrixing are two different items, and bracketing addresses exclusion of samples. It doesn't address the same thing as matrixing. In bracketing, if there is a range of strengths or a range of container fill or a range of container sizes, the idea is to use the extremes of those ranges and not test the interior samples, the mid samples. Test only the extreme samples. Matrixing, however, does not eliminate any samples. It instead eliminates testing at selected time points, so that not every product batch is tested at every time point, but the matrix design has to be chosen so that the product is tested across all of the time points, so that there can still be a profile generated for the product. Q1E is the data evaluation, and it's a discussion of the determination of retest period or shelf life estimation, and it's based on the accelerated, intermediate, long-term study results, and it describes the treatment of multiple batches, variable data, and statistical methods. The extrapolation, which is the part of the determination of the shelf life estimation, depends on the profile of the data and the extent of the data, the variability of the data trends at the various temperature conditions. You may be able to achieve either a 6 or a 12-month extension beyond the data that's available. For ANDAs, it's no more than 12 months. We have the SUPAC guidances again. These guidances are recommendations for product data and submission categories for certain post-approval changes. Basically, this is not a stability guidance. It's a much broader scope than the stability guidances, but it does describe batch scale, number of batches to be used in different conditions, depending on the type of change, and that number of batches may vary. It describes study length conditions, and it describes again the study commitment for long-term data. The final slide here are my final thoughts on this. I just wanted to go back for a second and say that we do care because we want to assure that the product maintains fitness for use, and that the effort put into justifying this is appropriate for demonstrating that use. The final thoughts: use the available guidance. The guidances define what is generally expected and might be acceptable and describe elements important to a successful stability program. Justify any deviations, because if you're deviating from what's generally accepted, you need to be able to support those deviations. Identify the relevant product characteristics, because having the relevant product characteristics is the basis for the program, for the results to be meaningful. And maintain the protocol in the program. Be faithful to it, because that's what generates the data used to evaluate the product. I hope that I've been useful and provided some insight into the tools and thinking that are useful in determining product stability. That concludes my presentation. Thank you for your attention.
Hello everyone. This is the last talk for this day 2 of the conference. And so far you've learned a lot. You've learned about the new drug application process, the abbreviated new drug application process, and some important aspects of the product like stability, and some programs like PEPFAR, etc. Now this talk is going to be focused on post-approval of a drug product. Once the drug is approved, the story does not end there. Many companies, that is when they start managing the production of the product beyond, and that's when the life of a drug begins. My talk will focus on the life cycle management of an approved drug product. This presentation outline will give you a short introduction, overview of the drug development process, and I'm going to define what a life cycle is, and then give you some unusual examples of how the life has deviated for some of these unusual drugs, and changes that can happen after an approval of a drug, and how they are based on risks, and some concluding remarks. Now, the pharmaceutical quality, just like the prior, you expect quality of anything you pay for and buy it in the market. Drugs are no different. So when you swallow a pill, you expect what the pill says it does to you, and you expect that what is there in the pill is supposed to be in it. So the patients expect safe and effective medicine with every dose they take. And the consistent quality should be maintained throughout the life of the drug product, and that is extremely important. Free of contamination and defects. It is what gives the patients the confidence in their next dose. I want to give you an example of our personal self. Supposing we are expecting a date on a particular day, and we have been looking forward to it for months, and this date has an important event in your life. Just before the date starts, you are getting a splitting headache. What do? So you go into the medicine cabinet, you pull out a bottle of ibuprofen or aspirin or acetaminophen or naproxen, any of the things which works for you, which you have it readily, you take it and swallow it with no questions asked. All you care about is getting rid of the headache so that you can move on with the next program in the agenda. That confidence with which you take the pill is what the agency provides. So in order to, you've seen in the previous talks about NDA, about different teams coming together to review a drug application, new drug application. Now, quality alone has different branches of different review teams coming together, because it is not one. It used to be that one person used to do everything, but now there are several teams playing a big role in approving the quality product alone. So the drug substance is one set of things, which is an active pharmaceutical ingredient. then the drug product, how it is designed, how it is manufactured, then the dissolution in biopharmaceuticals and bioavailability, then the manufacturing process, microbiological aspects of the drug, then the drug where it is manufactured, the facility and how it is inspected, whether what they say in the application is what is there happening on the site. All these things constitute a review process, and this review process is very involved, very thorough, and we make sure that the drug is of high quality. So to give you an overview of a drug development process, it starts from identifying a lead molecule, then you optimize the lead, and then take on to the different stages of development. The next two slides will give you an idea of what the process is all about. So the drug development process, discovery is the first step. Having found an entity, molecular entity, for a particular indication, we take on to the development of chemistry, biology, and the feasibility of the product, of a potential product. And this product will be determined what dosage form, what kind of delivery method, what kind of a treatment it is for, etc., etc. And the biology and feasibility are all worked out in the development stages. After this step, a non-clinical goes on. The non-clinical is toxicological testing on animals to make sure that the animals don't die by taking this medication, and this is tested, but it depends, and dosing is also tested on animals. Then there's the pre-IND stage. Pre-IND stage is a stage as an opportunity of overview of all the results you have and bringing it to the agency, asking for a meeting and to proceed ahead so that you can proceed ahead with some advice from the agency. So the pre-IND applications come, and these pre-IND applications are meeting packages, so where they ask questions and the agency advises them how to follow through. Then phase one is the investigational new drug, which is testing for safety of the drug product. The safety of the drug product is evaluated under phase one. Then under phase two, efficacy to some extent. And once the efficacy is feasible, it's the feasibility of efficacy, then as a large-scale efficacy is conducted under phase three. Once the phase three is complete, the results are compiled and submitted to the agency for an approval of an NDA. Right? At that point, that is when once the FDA has approved this application, the phase 4 starts. That's when the life of a drug begins. It is like having a baby. So the baby starts growing only after its birth. The main body of data for quality can be divided into major ones like the drug substance, drug product, packaging and stability, placebo information, and labeling. Placebo information is mainly applicable for investigational new drugs and clinical trials, but not for the drugs for post-marketing applications. Post-marketing applications are all real-time on the patient study. Next, how do you define a life cycle? Life cycle starts with the application of a new drug, right? Once the drug is approved, then it goes into a post-marketing setup. After the approval of a drug, that's when the real properties of this drug entity in the market, in patients, large populations, are identified and followed through. After several years of the drug being in the market, generic drugs come into play once patent expires or the exclusivity expires. Generic drug new applications come in after the fact. And then the post-marketing, then after a generic drug is approved, the post-approval of generic drugs come to play. The life of a generic drug begins. So the life goes on. So every company always tries to come up with new drug entities all the time, and they expect the approval of a drug product because they want the life of the drug to be long in the market. Something like ibuprofen or acetaminophen, etc., which has been there for decades and decades. I'm going to give some unusual examples of drug entities. One of the things is, from after a drug is approved, there's a lot of learning from the clinical trial which define the path of the drug. At the same time, the companies and the agency, we all learn as it is being consumed by the public. We get to know of events, serious adverse events, or the side effects, which are unintended consequences, all those things, and long-term consequences. All these things are learned during the post-marketing experience. And I'm going to give you some unusual examples, as I told you, in the next 4 slides. These 4 slides will show you only the structure of the active molecular entity, active moiety, and this entity will, just to tell you a story behind each one of them. So the first one, in the late 80s and the 90s, you had this drug called Seldane. Late 80s was approved, and this drug was called Seldane. The terfenadine is the active moiety, and it was actually for allergy. So this was about to become a blockbuster until they found out many of the patients who were taking it were getting arrhythmia. It really worked as an allergy drug. But when exploring the molecule further, why the arrhythmias were happening, they found this particular entity itself was very toxic. However, as soon as this drug enters into the body, it gets metabolized into a one of the methyl groups here, metabolized into a carboxylic acid, which is this entity here. Exactly the same molecule, which is a TOOH here. And it so happens that this metabolite is what is responsible for the antihistamine properties for allergies. So the company abruptly dropped this molecule and started developing this product. And so they pulled this out of the market after 13 years. But however, this molecule, brixafenadine, which was marketed as Allegra, became a blockbuster. And even today it's in the market, which is being used by many people. And there's had an enormous amount of public impact. Then another drug entity, Vioxx. You all know about this product. This is a COX-2 inhibitor, and it was in the market for 5 years. They pulled out from the market abruptly due to a lot of problems, adverse event problems, especially in cardiac adverse events in patients. The company voluntarily did it, and it is no longer available for that indication. So why am I giving this example? See, in the last example, really the entity which they discovered, spent so much money to discover, was not the correct entity, but that gave some unintended consequences of this metabolite being a drug entity, which is a blockbuster. In the same way, this was a blockbuster, but unfortunately, this life got terminated within 5 years because of adverse events, serious adverse events. Another drug has a different kind of a story, where this is minoxidil. Minoxidil is the name of the product, and this was originally developed for hypertension or high blood pressure. It worked better in growing hair than for hypertension. So they started developing this for hair growth after the clinical trial. So they used it as a topical cream or a foam or oil or an ointment, and this can be used for hair growth product as a cosmetic, which is a cosmetic thing. So cosmetic entity. So there's another example with Viagra. It's the same kind of idea of this was developed for high blood pressure. But when during the clinical trials they found out that it didn't work very well for high blood pressure, but the older people who were taking it were having very good sex lives. And that was the reason why they started focusing on this for erectile dysfunction based on the impact it had on the clinical trial. So this was approved for erectile dysfunction, and I was and became a blockbuster film. Why post-marketing changes, right? Why do you have to have changes? One of the things is the continuous improvement of the product. So until the drug is approved, the companies tend to spend minimal amount of time and energy and money to until they are sure that this is going to make a difference for their pocketbooks. So once it's approved, they know they can make money out of it. So the companies start working on improving, optimization, and focus towards marketing of the product. The continuous improvement is one of the aspects of it. Product optimization, incorporating of new technologies into the product, and process improvement. As I told you, it is like having a baby. Once the baby is born, then the baby learns. Every year the baby learns and grows. And that's exactly the way the drug, we all learn from the drug molecular entity, drug of a new molecular entity, the new molecular entity is what it does in the market. The process improvement is based on historical knowledge also. So over a period of time, they optimize to make sure this is consistently made to quality. And then regulatory requirements are there. So supposing during the approval process, they did not do certain things. They did not have any, let's say, stability data, adequate stability data. So they would be asked to commit to do certain experiments to make sure this entity, this drug product, follows through all the required guidances. And those comes under requirements and commitments. So in order to fulfill those requirements and commitments, the company does that work. And then the product quality issues, occasionally they arise. Stability can be a problem. Sometimes the formulation can be bad because it's not stable because of the formulation. Those kinds of issues can also have an impact, and that will make the companies do some improvement on product. And product, then business reasons. A lot of reasons is focus in the companies are toward business and economic reasons, supply and demand, etc. Next slide. There are different kinds of when the product is approved, you take up, then the changes start happening, right? You can make changes, or some products are so good they may make minimum changes. Or some companies ask the agency how to do this, how to do it. They come up with a meeting package and ask advice from the agency. There are three types of supplements we can do for changes. Efficacy supplements can come when there's for claiming a new indication in the product. It's safe. These are all some of the things I told you are unintended consequences. The changes in dosing regimen, safety changes. Sometimes safety is a problem, and black box warning may have to be issued. The contraindications with food, etc. Those are explored because not all these things are identified during the clinical trials. And then addition of dosing information for special population, for example, pediatric population or people with certain indications, certain diseases cannot take these, or contraindications cannot take this, or with certain food, dietary restrictions cannot take this, etc., etc. And sometimes nowadays we are going through the companion diagnostics. Some genetic, some genes have a problem of taking these kinds of drugs. That is also a part of today's added advantage of knowing the sequence and mutations. Labeling supplements. Labeling supplements changes to the labeling, which can be errors that are made during the original approval can be corrected, or constant improvement, and the medication guide, based on what is out in the field, how clear it is and what confusion it causes can be identified and made change, and changes can be made on those basis. Then CMC supplements, chemistry manufacturing control supplements, many kinds. I will go over it in detail. And changes to drug products, drug substance, manufacturing process, analytical changes, site changes, etc. Now, regulatory review times and clocks, I'm going to go over this. So the investigation of new drugs, I told you, when you are investigating phase 1, phase 2, when they apply for IND, within the 30 days, the agency has to review them and tell them that they are safe to proceed. We don't tell them, we don't approve these INDs. We simply say they are safe to proceed. In the case of NDAs, the standard clock is 10 months. And the priority, for example, drug entities which have a serious public impact, health impact, like and rare diseases impact, those get a priority application, like even oncological drugs get priority review clock, which is 6 months. In the case of post-approval supplemental changes, there are two types. One was a prior approval supplement and CBE 30 and CBE 0. Prior approval supplements are 4-month clock, which they cannot implement until the supplement is approved. In the case of CBE 30 and CBE 0, they can implement it 30 days after submission or 0 days after the submission, and the review clock is 6 months. And usually the onus is on the company and the risk they take for submitting CBE 0. The agency has a prerogative to change it to a prior approval supplement. Typical types of post-marketing application changes for a drug substance. What are those? A new manufacturing site. Many a times, a company will see a market for expanding. So they want to make more drug substance, and they may add a new manufacturing site. This manufacturing site will add more capacity to their existing product portfolio. And that is one of the changes. Then the other one is new supplier of regulatory starting materials. There are so many reasons why they go for a new manufacturing site or new suppliers. It may be the arrangements, business arrangements the companies make with different entities. Then the changes to routes of synthesis. This is very important because until the time that the drug entity shows promise that it's going to work in patients, they are not sure that it's going to be approved and it's going to be in the market. As you saw, some of the lives of some of the drugs abruptly ended too, even there. So the optimization is very important, and a shorter route of synthesis, better optimization, better yields, all these things are explored after an approval. And changes to the manufacturing process. Sometimes they have a batch process, and nowadays companies are exploring continuous manufacturing process, and that is one of the evolving, emerging technologies program that we have this continuous manufacturing process involved in, and we have a guidance for that as well. The changes in the process controls, like for example, or the drug substance specification. Supposing they identify a new impurity after approval, they have to report it and they have to make sure that it's qualified. So all these things come into play when they want to change the specification. So it can also be that they can add additional tests for specifications, and that would help assure the quality of the product. And changes to the shelf life. You learned a little bit about stability, how it matters, because everything has a shelf life, including us, and the retest period is very important. So we constantly monitor these things. The environmental elements like heat, light, moisture, humidity, and the atmosphere itself, which brings in the formation of many microbial organisms, etc. All these things matter for the shelf life of the drug substance and drug product. Now, a typical post-approval changes for drug products is represented in this slide. Again, there's a new manufacturing site. They may do it for changing or business reasons or expanding or scale up. Scale up is very important. So changes to the manufacturing process and or the equipment. Under the equipment, say for example, the manufacturing, if they want to change the size of the or scale up, they can make they have to make the equipment accordingly. So when they change the equipment, a lot of things within the process change: the speed with which they blend, the kind of granulations, timing, etc., etc. All those things are adjusted to. So that change should ensure that the ultimate product is within the specifications. And changes to the formulation is very important as well. Changes to the formulation can have a big impact on the bioavailability of product. Say they can go from an immediate release to an extended release, or this extended release can be over a period of 24 hours or 12 hours. And there can be many different scenarios depending upon the kind of excipients they use in the formulation. Sometimes formulation is changed for even to improve stability, and that is also an opportunity for better betterment of the product. Yeah, keeping up the quality. Then the changes to specification of the drug product, because as we learn, move along over time, and specifications matter. And if we add specifications, it's good to ensure the drug product quality, or changes to specifications are made, there should be adequate justification. Sometimes impurities are growing, they may expand the impurities, and the specifications are changed. As long as it does not impact the clinical outcome and the safety, then it should be okay. But these are all reviewed simultaneously when specifications change. So changes to the shelf life, extension or reduction of shelf life can be made. Companies volunteer to reduce if they find deviations in shelf life. And likewise, they submit a supplement for extension depending upon the products and the commitments they made during the NDA process. Introduction of new strengths is very important also, because originally when they develop, they are not, dosing is one of the most challenging aspects of drug development process. They have an optimal thing based on animal studies and clinical trials, etc., the impact, long-term impact, short-term impact, etc. But the developing new strengths will probably be based on what they see in the field. Say probably 5 milligrams, say that's the tablet, that's not enough. And they may develop it to a 10 milligrams and 20 milligrams, which are necessary for certain indications, and then patients as well. Same way, 10 milligram may be too much. They may want to bring it to 1 milligram because it's too narrow therapeutic index. So they want to change the product strength. Like the narrow therapeutic index is a very difficult thing to deal with. They have to titrate it from 1 milligram to several milligrams until they find the right amount of dosing that the patient can hold. So in order to do those kinds of titrations, there may be necessity for new presentations or new strengths. Okay. New presentation is something different. When a new presentation is probably like, for example, 50 tablets in one bottle and 100 tablets in that one bottle. So those are new presentations. Now, the regulatory basis of post-approval changes is under the law, it's a regulation, 31470, supplements and other changes to an approved application. The applicant must notify FDA about each change in each condition established in an approved application. Okay, so they have to, for an approved application, is used as a standard. They can improve on it, but they have to make sure and justify what the changes they are making are matching with the approved product, and it's going to have a positive clinical impact. The regulatory basis for FDA guidance. So, as I told you, the regulatory basis of the regulation, the CFR, regulation is the law. Based on the law, several guidances have come out, and these guidances for an approved NDA are listed here. Changes for an approved NDA, I'll show you how it looks. NDA or an ANDA. Scale up of post-approval change is called SUPAC, and it can be for immediate release, modified release, which is a sustained release, or semi-solid products. That's which is like gel caps or gelatin, which is a solid product, but at the same time liquid filled gels. Also, the manufacturing equipment addendum is another guidance. CMC post-approval manufacturing changes to be documented in annual report. And then comparability protocol is another guidance. I will talk a little bit more about it in the future slide. And pack analysis. If you want to, constantly companies do the testing outsourcing, and they change them depending upon the business contracts. So they go from one testing facility to another. Some testing facilities are limited by capability, so they find a capable facility to do these testing, the analytical testings. Now, this is the guidance page of the changes to an approved NDA. Is very important. You can look it up and get an idea of what it means, what are the different kinds of changes, or the different levels of changes, and what kind of a risk they are associated with. Just the same way, the regulatory basis is the International Conference on Harmonization guidances. Several countries have regulatory bodies which approve the drug to be used in those countries, just like the FDA for the United States, the European Medicines Agency for Europe, the British the British Medicines Agency for England and the British Isles, and then you have Japanese, you have Australian, etc. Many African countries have their own regulatory bodies. So in order to have a harmonized way of looking at it, these ICH guidelines keep coming up, and they are still in the process of developing more guidances to cover the entire spectrum of many changes that can happen. And these provide a basis for the companies to use them so that they can submit the supplements based on these guidances with the justification. They can follow, and everybody can accept. The regulatory basis for FDA guidance, some of the FDA guidances are unique, and since this is a body that has been for a longer time, we have guidances over, and that helps solidify, because we are still moving into the ICH realm, and that is going on. As it goes on, we have to abide by the FDA guidances. ICH Q12 is another guidance. That's the fundamental tools for enabling the support of harmonized lifecycle management. What is lifecycle management? So, in this guidance, it talks about established conditions and non-established conditions. There are two different things. Established conditions are the conditions that you can change for improvising the product or improvising or the quality, and you can plan and set. You can provide a plan to the agency, and the agency can approve it. And this is the post-approval change management protocol. Then pharmaceutical quality systems which Manage is basically the non-established conditions where everything is investigated during an inspection process. Established conditions are the ones which will change and can plan for it, and you can provide this plan to the agency for approval. Comparability protocol is a comprehensive plan. So I'll give you a typical example. If you want to change a manufacturing facility, what you can do is you can submit a protocol that you have planned to do this change to this facility, and that protocol will serve as a guideline for you to how you are going to implement the transfer. You can have the transfer done, and you can implement the transfer with the regulatory relief of CDER 30 supplement, and this will help you reduce the time of transfer, and the data can be reviewed after it is transferred to the new facility. So emerging technologies is another team in the agency where we encourage companies to come up with new techniques and new technologies. For example, I can give you a 3D printed tablet. That's a new thing. 3D printed tablet is a new technology, so how they're being done, and how this technology can maintain the quality of the product, same way as the batch process or any other process. And then guidance for industry, for continuous manufacturing. You can look up the guidance. I've given you the link here. Then I have told you about the changes, different kinds of changes. The major changes are to be submitted as a prior approval supplement, and everything is a case-by-case basis. These prior approval supplements are the supplements which you submit to the agency, and you cannot implement it until you get an approval from the agency. So the high impact to the quality of the product is one of so it has a high risk associated with it. You cannot be implemented. It cannot be implemented until it's approved. The review clock is 4 months. So it's the same way. Moderate changes, changes being effective in 30 days. That means you cannot implement it for 30 days, and after 30 days you can implement it at your own risk. These are moderate changes, and that 30 days gives the agency time to evaluate your submission to make sure it is moderate and not high risk or a low risk. So we have the prerogative to change it to a PAS or even CBE0, depending upon how the risk is, and we evaluate it based on a case-by-case basis. And the review clock is 6 months, and if you have to implement it within 30 days, it is at your own risk. So the minor changes sometimes the CBE0 or annual reportable supplements or annual reports are really low risk entities like editorial changes or etc. And which does not it does not impact the quality in any way. Can be implemented immediately after submission, and 6-month review clock, and the agency will decide if the company can go ahead with this or not. And everything is a case-by-case basis. Now, supposing you have a problem with your application and you want to make changes, you can come up to the agency and ask for questions. You put a meeting package together, depending upon the risk of the product, the necessity of the product in the market, the public impact the product has in the market. Type of meetings can be asked. Type A, which has a very high impact on the public health. Type B is moderate impact. Type C is low impact. And the Type A meetings are granted within 30 days, or actually the meeting should occur within 30 days. You have to come up with a package, ask specific questions, and the agency will respond back to you when the meeting is granted. For the Type B types of meetings, 60 days. So this is the best way to get a free consultancy from the agency. Type B meetings will be granted conducted within 60 days, and then type C meetings within the first 75 days. And everything is taken very seriously, and we give you appropriate responses for your questions, and you can meet with us and talk with us and deal with the changes, and you can get guidance from how to proceed further with your particular situation. So meetings are a helpful thing, and meetings are a part of the CFR, and you can always rely upon the agency's wisdom to get advice on your particular situation. Having said that, then I'm going to conclude saying that the post-approval changes are essential process improvement of the drug product, and this is important in many ways. So the drug life starts only after approval, and the constant improvements are made in this process, and this constant improvement is good for the quality, to maintain the quality. Then the patient response and the potential for repurposing happen during the post-approval phase 4 process. Then it all depends upon market demands and how big a product it is and how big a blockbuster it's going to be. Continuous improvement is essential to maintain the product quality and patient safety. Having said that, post-marketing is an important part of the review process, and we make sure that the product in the market is maintaining its quality to enhance or maintain the safety and efficacy and the patient welfare. Thank you very much for listening to this, and wish you well. If you have any questions, we'll all stand by.
Thank you so much for those informative presentations. We will now move on to our question and answer segment or panel. If you haven't had the chance to enter your question into the Q&A chat pod, please do so now, and we'll answer as many questions as time allows. But we may be having some connectivity issues for Dr. Holcomb at the moment, so we'll see if we can get those sorted out. But until then, we'll go ahead and start with some questions for Dr. Raghavachari. First question for you is, are bioequivalent generics, are they registered as NDAs?
Well, thank you, Nora, for the question. See, NDAs are new drug applications. Generics are ANDAs, abbreviated new drug applications. So there is in between called 505(b)(2). So 505(b)(1) is the NDA. B2 is the same molecular entity but with different say dosage forms, or sometimes the same dosage form comes as a different application. So bioequivalence is determined for a lot of reasons, right? So when the new drug is replicated as a generic drug, for an ANDA application, bioequivalence is required. Sometimes even within an NDA, when there is a new drug application and there is which they change the formulation, then there is a bioequivalence or a bio waiver is required. So there are two different issues here. Hopefully I've answered that question.
Yes, thank you so much for that response and making that distinction. We have another question for you. In which clinical phase will bioequivalence studies be considered?
This is a very nebulous question, but challenging one. So this is bioequivalence is for equivalency of the two different kinds of formulations or two different kinds of, say, from the NDA to an ANDA. So that's a distinguish. So bioequivalence is, say, for example, if you're changing the formulation, then a bioequivalence may be required. If you're coming from an innovator drug A generic drug comes up with a different formulation, then there's a bioequivalency is used to bridge the equivalency of the product.
All right, thank you for that information. Next, we have a comment and then question. I think FDA is working closely with industry on setting quality metrics. The question is on setting metrics around stability failure rate. Can you help with any references or guidance on setting metrics around this topic? Any industry practice or benchmark will help.
So, to the best of my knowledge, there is no standard because every stability issue is different from a molecular entity to entity and dosage form to dosage form. And it's a case by case situation. So, for just a run of the mill tablet kind of situation, there is probably upfront a lot of tests are done. Stability failures can be due to multiple reasons. So the root cause analysis has to be determined for that particular entity, and that has to be explored, and corrective actions need to be taken. To the best of my knowledge, there is no standard template with which we operate to determine the metrics of stability failure rate.
All right, thank you again for that information. We'll move on to another question for you. This is regarding your presentations. So regarding slides 21 through 22, how many batches and what type of stability data should be submitted for the typical post-approval CMC changes?
So it depends upon what is typical. So, for example, if you're changing the manufacturing site, right, you need minimum of you have to have 3 batches, and depending upon the dosage form, like for example, if it's extended release or immediate release, if you have the history of it, everything, as I said, is a case by case basis. So we might want to decide, and then it's also the trends. It depends upon the trends. If you have usually it's about 3 batches and 6-month data is required, but the agency is flexible about how you justify or how much you can provide with the commitment. So there is a wiggle room there for you to work with.
Okay, thank you so much, Dr. Raghavachari. So it looks like we might have Dr. Holcomb available now. So we'll try first question for you, Dr. Holcomb. Are you aware of current and reputable stability tests and or literature that have been performed on herbal medicinal substances, specifically cannabis inflorescence?
That's an easy answer because the answer is no. I'm not aware of any consolidated resource on that. So I would suggest just simply using what literature was available, because there will be very little, if any, guidance from the FDA on cannabis.
Thank you so much. We're glad that you're able to come online and provide that information for us. We have another question for you. If bracketing is applied, does the agency expect that all samples and strengths, fill volumes are stored but not sent for analysis, or only samples that are planned to be tested can be stored in stability chambers?
That's a good question because you might think, well, I might want to do something else with the product that I have. But the stability protocol describes what testing will be done and under what conditions. So the stability protocol is a bracketing protocol that calls for testing at the extreme ranges of the product configurations. Then there is no reason to include the center product in the stability chambers because they will not be tested. If you believe that there may be some situation in which you want the additional information, then of course, that's a decision to be made outside of the protocol. But no, the specific question was, do you have to store everything if you're only going to do the extremes on bracketing? And the answer is no.
Thank you for that response. We have another question for you, Dr. Holcomb. Are the available resources for formulators or scientists to utilize in order to ensure all required guidelines, i.e. ICH, USP, or SUPAC, are utilized prior to an FDA submission?
I think Ramesh's slides have a very good resource discussion. And there is, to my knowledge, nothing that says here are the guidances that you have to use to make sure that you've gotten them all. What you need to do is you need to access the ICH guidances, you need to access the USP to look at monographs and specifications and the general chapters, and you need to use the web link that Ramesh provided for the FDA guidances. There is nothing to point you to, here's a guidance you need. There, however, are a large number of guidances that are available, and those should be searched in order to find out whether your particular product is applicable or the requirements of the guidances are applicable for your product.
Thank you again for that response. So now we'll move back to some questions for Dr. Raghavachari. Next question is, could you please provide more clarifications on ICH Q3D guidelines for impurities and applications?
I got lost. Oh, okay. Yeah. The ICH-Q3 is impurities. It tells you about the qualification threshold depending upon the unknown impurities and known impurities. So it all depends upon then we work with if the quantity is beyond a particular limit of the, say, I think it's 0.5, or you have to check the guidance exactly depending upon what the amount is. So if it's beyond, say, it also depends upon the per day daily dose. And then we work with the farm talks folks to see if it is necessary that it needs to be qualified or not. Now, this works in combination with the in silico toxicological methods, and then farm talks folks what they feel is important, and then they may ask the applicant to qualify those impurities. So that's the way it works. So it all, again, it's a case-by-case basis, and how well your analytical methods, it depends upon the limits of quantitation and limits of detection and the method used. So it's a combination of several things play into role. And when you say clarification, I'm assuming that this is what you want to know, whether it's all set in stone. No, it's not, because I think it has to work. Multiple moving parts are involved in this. Hopefully that answers the question. It's not a direct answer, but it's a difficult.
All right, thank you for that information. That's very informative. We'll move on to another question for you. Would changes to packaging size with no modification to a product require a post-approval CMC?
Yes, that answer is pretty straightforward, because there are two things. When you have a new presentation, I'm assuming that you're coming up with a new presentation with no changes to the drug product, right? But then this has to go into the label. It becomes a labeling supplement as well, and a new presentation. So it has to be documented, and labeling has to change. So you have to submit a CBE 30 supplement for that.
All right, thank you. One more question for you, Dr. Raghavachari. For the drug products under drug shortage, can the sponsor request expedited review? If yes, what's the typical timelines for PAS and CB 30 supplements?
So for a drug shortage situation, we work with the drug shortage group. And we would make sure that it really does have a public impact, public health impact. So in such a case, you can request an expedited review, and we do take care of it at the upfront, um, as soon as it arrives and alerted. So normally we expect to get things done depending upon what is needed for the approval or review of that particular application. It depends again. So if it does not involve, say, facilities inspection, so then one of the things is facilities inspection may slow down the process, because you have to schedule an inspection, and then when it is scheduled, we don't know. So depending upon whether it's a domestic site or a foreign site. So a lot of those factors play an important role. But in general, yes, you can request an expedited review, and we will do everything we can to expedite this review. And normally, prior approval supplement is 4-month clock. But for a CBE 30 supplement, it's a 6-month clock. And that's when the approval has to be, provided, or action has to be taken before that time limits. But then, although if it's a drug shortage thing, when it depends upon, say, an inspection of a facility, and the facility is not in a good shape, then there are other alternatives we need to explore. Hopefully that answers your question.
All right. Thank you so much for that response. We'll see if we have time for one more question. This would be for Dr. Holcomb. What is the post-approval stability commitment for?
The post-approval stability commitment was added into the ICH Q1A because the requirements for in Q1A don't require that the market package be part of the approval process. So you might have the smaller batch sizes in your application, and that's what the data is from to generate the approval, along with a comparison of the proposed larger scale. So in order to get information on the actual product that's being manufactured at the scale of commercial manufacture, the post-approval stability commitment has become, and I'll use the word requirement, in order to connect, if you will, the actual market product with the product data that was used to evaluate and approve the product. A secondary use is that in one of my slides, I talked about a quality attribute as stability being an attribute. And so if you think of it that way, post-approval stability gives you an ongoing life cycle kind of idea of whether or not you're getting drift in your manufacturing or in your raw materials, because you continue over some period of time, only on one batch a year usually, to see whether or not you're maintaining the same quality attributes that you were during the time of approval.
All right. Thank you so much. And that is unfortunately all the time we have for questions right now. So thanks again to Dr. Holcomb and Dr. Gavachari, and again to all our presenters for the excellent information that was shared throughout today.